Hi everyone, I'm Chris Probert, and I'm here to tell you about our work on learning determinants of nucleosome positioning through sequence-based models of cell-free DNA. Before I start, I want to acknowledge that this is joint work with several folks from Anshul Kandaji and Christina Curtis's labs at Stanford, as well as from Julia Zeitlinger's lab at the Stowers Institute for Medical Research. The genome exists in cells as a three-dimensional complex called chromatin, and I'd argue that nucleosomes are really the most fundamental unit of that structure. Individual nucleosomes themselves are octameric protein complexes composed of individual histones. They're cylindrical in shape, and they're roughly 11 nanometers in diameter. The image on the right is data from a molecular dynamics simulation of DNA sequence wrapped around a nucleosome. And you can see that the core sequence itself is fairly stationary, whereas the flanker or linking sequences that come off the edges of the nucleosome are much more free to move around during the simulation. You can also see that the blue cylinder that goes through the nucleosome is an axis of symmetry, and that's what underlies a lot of the symmetries that we'll see in the sequence basis of nucleosome positioning. People have previously done a lot of work in studying sequence basis of nucleosome positioning. The main effects are thought to break down into two main categories, the first class being intrinsic effects. So intrinsic effects are the relationship between the actual sequence that a nucleosome is bound to and the nucleosome occupancy. So you can see on the left that there are certain dinucleotides that are much more prevalent within core sequences of nucleosomes versus in flanking. And then on the right, you can also see that there's periodistic patterns that relate to the major groove of DNA and certain residues on the solvent-exposed surface of nucleosomes having preference for interaction with different classes of dinucleotides. The second major component of DNA sequence effects on nucleosome position are called extrinsic effects. These are driven by proteins such as transcription factors. Here's an example from the yeast genome, where you can see there's a Gal4 site that's displacing nucleosomes at its binding location, but it's also positioning flanking nucleosomes that extend away from the TF site. So this is kind of how transcription factor binding has long-range extrinsic effects on nucleosome position. There's several methods available for measuring nucleosome position in the genome, most canonical of which is MNase-seq. MNase works by degrading unprotected DNA sequences and then sequencing the nucleosome-bound fragments. Another method is ATAC-seq, which works by transposing sequencing adapters onto open chromatin, essentially identifying locations of nucleosome boundaries, and this gives a very precise readout of nucleosome position at active open chromatin elements. Third approach is chemical mapping, which is of these three the most precise, and this works by inserting germline edits of histones H1 or H4, and then performing a chemical conversion of the actual DNA base that is touching the center of the nucleosome, and this provides a precise readout of the exact nucleosome position, but it does require a germline edit, so it's not tractable in humans. Another approach that's been described for measuring nucleosome position is cell-free DNA sequencing. Cell-free DNA is fragments of DNA that are prevalent in blood plasma and mainly coming from blood cells or other cell types in the body as they apoptose, and they're typically protected by proteins, either nucleosomes or TFs, because otherwise the DNA fragments are degraded quickly. It's been shown that it's possible to extract nucleosome occupancy scores from cell-free DNA alignment matrices, and there's also a fun historical basis for the original discovery of cell-free DNA from the single ladder gel image in a single author publication in 1970, where the author actually also correctly predicted the ladder structure of nucleosomes around DNA. So the first thing we wanted to do was see if we could train an optimized detector of nucleosome occupancy from cell-free DNA alignment matrices. We designed a two-step alternating EOM-like optimization procedure, which we call the nuc-D or nucleosome detector. Works by initializing the filter from nucleosomes that are extracted from CTCF sites, since they're sources of well-positioned nucleosomes. And then we perform alternating rounds of steps where we scan the genome with our current estimate of the nucleosome density filter. This gives us a nucleosome signal, which we can call peaks from, and then we can consolidate those peaks to update our filter. We basically repeat this procedure for something like 20 or 30 iterations until we converge on a filter, which is the filter that's shown at the top. To validate the nuc-D filter, we compared to MNase sequencing and showed that we got very consistent locations of nucleosome calls, as well as the relative intensity of the signal across nucleosomes around different types of regulatory elements. We also compared to other filters that have been previously described for measuring nucleosome occupancy from cell-free DNA using a metric we call the proportion of spectral energy, which is just measuring how specific the filter is for nucleosome frequency waveband. And we show that the NucT filter significantly outperforms other hand-designed filters for nucleosome occupancy measurement from cell-free DNA. So now that we've got this high-quality nucleosome label, we wanted to train a model to predict it from sequence. So we designed a convolutional neural net architecture for this task, which takes as input one hot-encoded DNA sequence of length 5,000 base pairs. And then there are 3 dense convolution layers at the beginning, which are designed to detect intrinsic effects. And they work for this purpose because they're dense, so they are able to examine, learn sequence relationships that span within an individual nucleosome. And they also have a receptive field that is larger than a mononucleosome. So this is where the model, we think, is designed to learn intrinsic effects. And then on top of that, there are a series of 11 dilated convolution layers, which are scanning successively larger and larger sequence contexts. And these are really designed to try and detect extrinsic effects, so longer range sequence factors that are driving nucleosome positioning. The model outputs a single channeled, which is the predicted nucleosome occupancy, and we score it against the true NucT label using negative multinomial likelihood. Here's 4 example regions from held out test chromosomes. And you can see that we're achieving pretty good predictive accuracy for the true nucleosome density signal. The overall Pearson correlation, the median Pearson correlation on these 2,000 base pair bins for held out test chromosomes is 0.82. So now that we've got this fairly high accuracy model, we wanted to really ask what are the sequence elements that are driving its predictions. First thing we did was develop a custom, essentially feature ablation-based interpretation method, which we call the shuffle mutagenesis score, SMS. This is designed to detect DNA sequence elements that are having long-range extrinsic effects on nucleosome positioning. It works by sliding a 9 base pair dinucleotide preserving shuffle across the genome, essentially shuffling 9 base pairs at a time. For each location where we apply a shuffle, we calculate the difference from the reference prediction, essentially the effect of mutating this 9 base pair sequence. And this gives us a 2D matrix of the location where the mutation was applied versus the response across the output window. We then take a Fourier transform of the effect of each position, and this gives us a readout of how widespread and how periodistic the spectral energy of the mutation in each position. So this really pinpoints locations that are having widespread extrinsic effects. So to help give some intuition for what's actually going on in the SMS score, here's a short video where we're scanning in yellow. We're applying the SMS mutation across a region flanking a CTCF site, which is in the center. As we get to the CTCF site, you'll see we suddenly have a huge mutation effect right there. And then as we move away again into flanking sequences, the 9 base pair shuffle is really not having much effect at all, only kind of local disruptions to the predicted nucleosome occupancy. So here's a vignette from a sample region on chromosome 19, where there's a CTCF rest site, and you can see that the SMS score is really highlighting the causal sequences under the transcription factor motifs that's driving the extrinsic positioning at this locus. And zoom in at the bottom, we're comparing the SMS score against another feature interpretation method from the same model, DeepShap, which is based on backpropagation. And you can see that both are highlighting the core sequence that's underlying the transcription factor binding sites, but the SMS score has actually better segmentation of the core sequences than the DeepShap, which is a little noisier within the TF motifs themselves. Validated SMS also more systematically on 10,000 SPI1 sites, comparing SMS score to a different formulation of SMS score with an independent sequence shuffle, getting very good rank correlation. And then for 10,000 CTCF sites, we compare the SMS score against the deep SHAP score, and again, get very good consistency. So now that we've got this method for measuring the extrinsic sequence basis, we wanted to look at a bunch of different transcription factors and see what are their blast radius. So essentially what we're doing is we're taking 1,000 sites for each TF, and we're ablating the sequence motif that underlies the site and measuring what is the effect on flanking nucleosomes. Not surprisingly, CTCF has by far the largest effect, and it's got a very large blast radius in flanking nucleosome space. Mutating the CTCF motif really displaces the flanking nucleosomes. Other chromatin regulators like YY1, SPI1 also have fairly large blast radius, and then as you move into TFs that are playing less of a chromatin regulatory role, the blast is much less pronounced. So we can also measure this. Essentially, the top metric here, or the motif log fold change, is the motif hypersensitivity. The bottom is the blast radius, the width of the disruption envelope. And you can see that we are able to sort of stratify TFs based on their influence they're having within their binding contexts on flanking nucleosomes. So now that we've kind of measured these extrinsic effects, we wanted to see how do they exist in balance with intrinsic effects. One of the models that we trained while developing DeepNuke was a variant of the model where instead of taking one hot encoded DNA sequence, it takes in smooth dinucleotide features. So these are the densities of each of 16 possible dinucleotides that have been slightly smoothed with a Gaussian filter in order to mask the underlying sequences themselves so that the model can't actually learn TF motifs, but it can still see the coarse grain electrostatics that underlie intrinsic positioning. So on the left, we're showing for reference sequence, a CTCF site, and on the right, the same CTCF site, but with the actual CTCF motif ablated. And we're showing in orange the prediction of the DeepNuke-seq model, and in green the prediction of the DeepNuke-dinuke model. And you can see that under the reference sequence, the DeepNuke-seq model aligns very closely with the true cfDNA, whereas upon motif ablation, the DeepNuke-seq model actually now much more closely resembles the DeepNuke-dinuke model. And so what this is suggesting is that the correlation between the difference in correlation between reference sequence and with motif ablated between DeepNuke-seq and DeepNuke-dinuke can be used as a measure of how strongly the intrinsic versus extrinsic effects were leading to nucleosome positioning in a given context. So we can take this approach and apply it across instances of different TFs. And essentially what we're doing is measuring the change in correlation between DeepNuke-seq and DeepNuke-dinuke before and after motif ablation. And so the effect size and the p-value of the rank change are essentially measuring the strength of the difference, the strength of the difference between the extrinsic and intrinsic effects that are revealed upon motif ablation. So now that we've looked at the relationship between extrinsic and intrinsic determinants, we wanted to go and survey intrinsic effects themselves. So on the left, we're showing the dinucleotide constraints. So this is taking nucD signal, estimating the nucleosome dyad locations, and then calculating the log2 of observed over expected sequence prevalence of each dinucleotide at each position. And you can see that there's kind of strong spatial patterning of different dinucleotides. And you can also see that there's reverse complementarity expressed. So you can see that the CC, which is the reverse complement of GG, is symmetric about the dyad as we move, away from the dyad location. We see kind of this exact mirror imaging, and this holds up for all other reverse complement dinucleotides. On the right, we're showing a different metric, which is based on the actual model predictions. It's called expected disruption score. So this is essentially the effect of injecting each dinucleotide into each position across a bunch of different 10,000 nucleosome instances. And we're basically measuring what is the effect of inserting this dinucleotide on overall nucleosome occupancy. And you can see that the spatial patterning of the essentially mutation effect matrix closely aligns with the spatial patterning of the dinucleotide constraint matrix, although the mutation effect matrix is overall more negative, which is shown by the scores in the bottom, which are essentially the marginal sums of each position across all dinucleotides. What this is suggesting is that there's actually higher order sequence contexts, so k larger than 2, that are driving actual observed nucleosome positions. And finally, one last thing to end on is we're also able to measure epistatic interactions between non-adjacent bases that are having influence on nucleosome occupancy. So what we're doing here is the same as the previous slide, except instead of just substituting a single base or a single dinucleotide into each position, we're substituting all possible pairs of bases into all possible pairs of positions. And we can ask, what is the difference between the full observed effect under the model of inserting these bases into these positions versus the additive expected effect, because we can also score, we also know the marginal contributions of each, or the marginal effects of each base into each position. And so the difference of the observed and the additive gives us these interaction terms, or in genetic speak, epistatic effects between different bases. And we see strong off-diagonal banding with 10.5 base pair periodicity, which suggests that the sequence is having effects that are propagated across the major groove of the DNA as it wraps around the nucleosomes. So with that, I want to wrap up and thank everybody who's been involved in this project from the Kondaji, Curtis, and Zeitlinger labs, and thank you all for listening.